This is the second portion of the blood lecture. We're going to begin with white blood cells and finish up with talking about red blood cells and antigens. So, when you think about white blood cells, I want you to think about them in terms of being our body's defense system. Another name for white blood cells is leukocytes. So, you'll sometimes hear about something called leukocytosis, and that just means an elevation in the white blood cell count. There's another term called leukopenia, and this just means a decrease in the amount of white blood cells. Some characteristics of white blood cells is they have a nuclei. This is different from the red blood cell. Remember, they lack a nucleus. They do not contain hemoglobin. White blood cells are a complete separate entity. They do not carry oxygen. They're defined by the presence of granules. They're either granular or agranular based on the staining and highlight of these granules, as you'll see in the next picture. The granular leukocytes are the following. They're the neutrophils. These are the cells that are going to be responsible for attacking bacteria whenever you get an infection. The eosinophils will stain red. That's important to know. The eosinophils are responsible for attacking parasites. And then you have the basophils, which are important for some allergic reactions. And then you have your agranular leukocytes. These don't have granules. These are your lymphocytes and your monocytes. Lymphocytes will be important for immunity. They'll be important for stimulating memory cells whenever you receive a vaccine. Monocytes are also phagocytic cells that differentiate into macrophages once they come out of the tissue. This next slide, you'll see the presence of granules in the eosinophils. Again, this is going to have a red appearance. This is a way that you can identify an eosinophil on a slide. The neutrophil, you know that it's a neutrophil by the presence of the distinct lobules. There's 1, 2, 3, and 4 lobules present here. And then the basophil has this deep purple appearance. The lymphocyte has this classic appearance here, and the monocyte has this bean-shaped appearance. So again, as you can see, there's no granules present within the lymphocyte and the monocyte. However, there are the presence of granules within the neutrophil, within the basophil you can see here, and within the eosinophil. Again, this is just another picture here kind of depicting, showing you the monocyte here, neutrophil having the different lobules, the eosinophil is red, and this basophil is the deep purple. So what is the function of the white blood cell? General function is going to be phagocytosis. Remember, phagocytosis is just an invagination of a cell membrane and taking in a solid particle. So this could be for an antigen that comes in. This could be in the form of a vaccine. That's how vaccinations work, is you introduce an inactive antigen and your body takes it in and then stimulates an immune response. Opsonization, this is going to be this is essentially just something called neutralization, neutralizing the bacteria and not allowing the harmful effects to occur. It's important to note that white blood cells are far numerous than red blood cells. Far less numerous than red blood cells. So red blood cells are going to be primarily the most abundant within the body, and white blood cells are typically between 4 to 10,000. This here is a process known as diapedesis, or just immigration of the white blood cells. It occurs due to a response called chemotaxis. This is just, it's like a chemical taxi, basically calling the white blood cell to come out of the capillary and migrate to the area of inflammation. So we'll read the text here. Many white blood cells leave the bloodstream in response to bacteria, viruses, or harmful chemicals. So you've got this guy outside, and he's just causing a lot of trouble. I want you to think about it being in a neighborhood. He's just really loud, causing a lot of trouble, and essentially all the neighbors come out of their house and migrate down the street to find this guy and see what he's doing. That's kind of what is happening here. So they roll along the endothelium of the cell, and the endothelium is just the inner lining of the red blood cell. They stick to and squeeze between the endothelial cells. So rolling along the endothelium, this is called margination. And then when they actually squeeze between the endothelium, this is called diapedesis. The precise signals vary for different types of white blood cells. It depends on the interleukins that are present, which are the chemical messengers of the cells. We talked about that in Bio 137. This is just a picture here showing you what's actually happening. This is a neutrophil. Again, a neutrophil is going to be one of the white blood cells. You can tell by its different lobules here. And it is, there's some type of bacteria or some type of antigen out here causing the neutrophil to want to leave the blood vessel to get to the area of injury or the area where the antigen's present. So you've got the endothelial cell. Rolling is happening here. It actually rolls across the endothelium. There's these little slits within inside the capillaries, and it's going to squeeze out of the endothelial cell to get to the site of action. We'll talk about these in particular now. I want you to know that the neutrophils and the macrophages are the primary phagocytes, the phagocytes of the immune system. And what does that mean, phagocytes? So they essentially just engulf foreign particles. So the neutrophil and the macrophage. It's important to know that the neutrophil is the white blood cell that is the first to the site of injury or inflammation. The neutrophil is going to be the quickest responder. So it's essentially like your ambulance. You know, you call 911. He's the ambulance. He's there the first. He's there at the beginning. So neutrophils respond mostly to damage by bacteria. So if you go in and you've got a bacterial sinus infection, you can expect to see an elevation in the neutrophil count. Neutrophils are, again, we haven't got into this, but whenever we cover our immune lecture, we'll talk about something called innate, which is the first line of defense. This line of defense is, there's no memory here. This is just your body's natural ability to fight off foreign antigens, right? Foreign, like a cold, your common cold. This is your innate immune system. When we talk about memory, we think about vaccinations, and this is going to be your body's secondary line of immune system. We think about vaccinations. We think about maybe we've had a cold once, and our innate immune system responds to it. It's primarily through the neutrophils and the macrophages. But later on, if we develop a re-exposure to this, we have memory of this now. Our first encounter caused us to maybe form something called memory cells, which will be part of the second line of defense. But again, we'll cover this at a later topic or at a later time. Monocytes, these take longer to arrive, but they arrive in larger numbers and destroy more microbes. They are called monocytes when they're in the tissue, but once they leave and enter into the blood, or leave the blood, they're called macrophages. Basophils contain granules. Again, we've covered this previously. They contain different, they release different hormones, heparin, histamine, and serotonin as sites of inflammation. They are or play a role in some hypersensitivity reactions, some allergic reactions that occur. They're involved, or they intensify inflammatory reactions. What I want you to know primarily on this slide is that eosinophils, the white blood cells that stain that deep red appearance, they are responsible for releasing histamine, and they work against certain parasites. So you have to think, what is a parasite? Well, whenever you think about a parasite, I want you to think about something that comes into the body and causes harm. This could be, for example, a hookworm. There's several hookworms that live in South America. Let's say you're walking on a beach and a hookworm penetrates your foot and gets into your body. Well, the eosinophils are responsible for identifying this parasite and destroying it. Here's some selected videos that could be important for you to kind of look at and just kind of hone this in. Now, I talked about the innate immune system. Now I want to kind of talk to you about the secondary line, our memory, our body's ability to remember things. These are the lymphocytes. They're the major soldiers of the immune system. You have B cells and T cells. The B cells are responsible for destroying bacteria and inactivating toxins, something called opsonization. This is neutralization of a foreign antigen. T cells, they're responsible for attacking viruses and funguses and some cancer cells. In HIV, this is, remember, human immunodeficiency virus. This is when T cells are destroyed, something called CD4 cells, CD4 helper cells. And this is when T cells are destroyed. So you're more prone to infection from viruses and funguses because of that. For example, if you had an immunodeficiency in which your B cells were destroyed, bacteria, you'd be more prone to bacterial infections. Now we'll talk about something called platelets or thrombocytes. That's another word for it. Platelets form, again, from the pluripotent stem cell, again, through the myeloid lineage. If you can remember, the myeloid stem cells give rise to something called the megakaryocytes. And the megakaryocytes fragment into things called platelets. And platelets are responsible for blood clotting. So again, you have the myeloid stem cell, or you have the pluripotent stem cell present in the bone marrow. It gives rise to the megakaryocyte, and the megakaryocyte will fragment and will then form platelets. So each of these fragments are known as platelets. Platelets are responsible for hemostasis. Hemo means blood, so causing blood clotting, leading to the formation of blood clot. So like red blood cells, platelets also have no nucleus. So why would we need to form a blood clot? Well, if you cut your finger, right? If you have some type of trauma in which you have internal bleeding, we have to have a built-in mechanism in order to stop this bleeding. So not all blood clots are pathological, or it just depends on the body's ability to break down these clots. So this is here. If you put this in PowerPoint mode, in which I can't in order to record this lecture, you'll see the formation of something we call the platelet plug. And we'll describe this in more detail. The first step of platelet plug formation is platelet adhesion. There's something called von Willebrand factor, which is responsible for binding to a molecule and on the endothelium. After the platelet adheses, it will again release some inflammatory reactions here, and then eventually we'll have something called platelet aggregation, in which the platelets come together and form something called a platelet plug. Just want you to know these steps. First, you have platelet adhesion, then you have release of chemicals, and then you have platelet aggregation to form a platelet plug. So hemostasis, again, this is just a sequence of responses that stops bleeding. There's three main mechanisms in which this happens. First, the blood vessel, whenever there's trauma to it, it's going to contract. This is called a vascular spasm. So the smooth muscle present in the artery or the arteriole is going to contract. After this, it's going to trigger the platelet adhesion to occur. You're going to release the chemical mediators, and then aggregation is going to occur. This is going to then form the platelet plug, and then we're going to have something called the coagulation cascade. So there's something called primary hemostasis and secondary hemostasis. In primary hemostasis, you have formation of the platelet plug. So this is when adhesion, release of the chemical mediators, and platelet aggregation occurs in order to form the platelet plug. So this is kind of like the first line. This platelet plug is not as strong. It doesn't actually form a strong meshwork. You just have this weak platelet plug. But in order to make the clot strong, we then have to have the coagulation cascade. And I'll describe what that is on this next slide. So blood clotting through the coagulation cascade. We'll first describe blood clotting. So this is clotting is a series of chemical reactions culminating in the formation of fibrin threads. So there's several different enzymes or factors that are present. Calcium has to be present. There's several inactive enzymes that have to first become activated through this cascade. And you'll see here, this is the formation of early clotting. As you can see here in the intermediate stage, it becomes a little bit more complex. And then eventually here you can see this fiber meshwork here. This is the red blood cell, and then the whole purpose is to form this strong fiber meshwork. We have the extrinsic or the intrinsic pathway, leads to the formation of something called prothrombinase. The whole purpose of prothrombinase is, again, to convert prothrombin into thrombin. So thrombin is going to convert fibrinogen into fibrin, and we'll see that. I just want you to see that here the major point is that at either end of the pathway, this is going to form threads that create the clot. This picture here just kind of depicts what we were talking about. The extrinsic pathway and the intrinsic pathway. The intrinsic pathway is going to take a little bit longer. It's in direct contact with the endothelial cells, whereas the extrinsic pathway has something called tissue factor, which causes the activation of factor 10, which converts prothrombinase into thrombin. Okay. So the extrinsic pathway and the intrinsic pathway. The extrinsic pathway, tissue factor or thromboplastin leaks into the blood from cells outside or extrinsic to blood vessels, initiates the formation of prothrombinase. But in the intrinsic pathway, it's directly in contact with blood. So again, the extrinsic pathway, fewer steps than the intrinsic, and it occurs rapidly. The intrinsic is more complex and slower than the extrinsic. Tissue factor is going to be responsible for activation of prothrombinase, whereas in the intrinsic pathway, the outside damage is not needed. So this is just a comparison of the two different pathways. Here's some videos that'll kind of help identify with blood clotting. We'll kind of finish here with talking about blood groups. So depending on which antigen is present will depend on what blood type you have. If antigen A is present, you'll have blood type A. If antigen B is present, you'll have blood type B. If no antigen's present, then you'll have blood type O, and you're considered the universal donor. If blood type A is present, you have antigen A, which means you don't have antigen B, so you form antibodies against antigen B. You'll have anti-B antibodies. If you receive B blood, you have the antibodies to the B blood, so your body will attack B blood, and you'll have a transfusion reaction. So again, just to kind of go through this again, depending on the presence or absence of the antigen on your red blood cell will determine what blood type you are. If you have antigen A, you're blood type A. If you had antigen B, you're blood type B. If you have neither antigen, then you're the blood type O. You're the universal donor. You can donate to anybody. If you have antigen A present on your blood, then you have antibodies to B. If you receive B blood, then you have antibodies to that B blood. Therefore, your body will attack that, and you will undergo a transfusion reaction. This is why whenever you come into the hospital, they'll do, if they expect you to need blood, they'll do something called a type and cross, where they'll take your blood type, they'll cross it to figure out which type that you are, and in case you need that blood, type A, they'll only give you A blood. This is a picture here just kind of depicting what we went over. Antigen A, antibody to B. Antigen B, antibody to A. Both A and B have neither antibody. So they're the universal recipient. They can receive any type of blood. They can donate to anybody, but they can receive any type because they have neither antibody. This is talking about the Rh group. The Rh group is just the presence of the D antigen. The D antigen. So if you have the presence of the Rh group, you're Rh positive. If you have absence of the Rh group, you're Rh negative. So if I'm A positive, that means I have antigen A and the Rh antigen present on my blood, right? So this is describing something called hemolytic disease of the newborn. People whose red blood cells have the Rh antigen are Rh positive. We just said that. People who lack the Rh antigen are Rh negative. So hemolytic disease of the newborn is a disease which happens in which the mom is Rh hemolytic disease of the newborn, if the blood from the Rh positive fetus, so the mom has a baby that is Rh positive, it has the Rh antigen, the rhesus antigen present, but the mom's Rh negative. Okay, the mom is Rh negative; she doesn't have it. This causes the formation of um, anti-Rh antibodies. Right, her blood is in contact with it. This won't affect her the first time, but however, she has another baby who is Rh positive, and this can cause the mom to undergo hemolysis of her red blood cells. This is a difficult concept that we'll kind of discuss in class at a more detailed um, description. But again, I just kind of wanted you guys to be familiar with this. This is an Rh negative mother with an Rh positive baby. Okay. Um, because the baby's Rh positive, her body, right, she's going to form antibodies to the Rh antigen, right? She has another baby that is Rh positive. This will undergo hemolysis. There's a um, there's a shot that we can give these women called the RhoGAM shot, in which it's essentially just anti-Rh antigens that we give this to these women in order to prevent hemolytic disease of the newborn. This is just describing to you agglutination. Agglutination is just the clumping of blood. This is what we use in order to determine which blood type you are. So again, guys, look over the objectives that I put on the handout. We'll discuss this in more detail in class. If you guys have any questions, please feel free to reach out to me. Thank you so much.